Good evening, Veritas Vanguard interns. Am I audible?
Yes,
Yes,
sir.
anyone? Yes, can anyone please confirm if I'm audible?
Yes,
Yes,
you
you
are.
are.
Okay. So, welcome to one of the most scientifically dense and fascinating sessions of our internship. Today, we are exploring the triangle of evidence that is, forensic biology, toxicology, and forensic medicine. To cover this vast spectrum, we have power-packed panel of three experts who bring experience from prestigious universities, laboratories, and top-tier medical institutions. First, to guide us through the microscopic world of biological evidence, please welcome Ms. Priya Varshini S. Ms. Priya is a specialized biotechnologist and forensic scientist holding an MSc in forensic science from the prestigious National Forensic Sciences University, NFSU, Greater Noida. Her expertise lies in the heart of the laboratory. She has served as a training forensic assistant at the State Forensic Science Laboratory, where she gained rigorous hands on experience in DNA extraction, isolation, and serological techniques. Her ability to handle complex biological samples is backed by her research on the quantitative extraction and analysis of DNA from trace saliva evidence, a critical skill for solving crimes where evidence is invisible to the naked eye. With a strong foundation in biotechnology from the University of Madras, she bridges the gap between classic serology and modern molecular biology. Please welcome Ms. Priya Varshini S. Next, for forensic toxicology and medicine, we would be introducing Ms. Jyoti Chauhan. Ms. Jyoti is a toxicology specialist currently pursuing her master's at Jamia Hamdard University, New Delhi. What makes her profile unique is her direct exposure to forensic medicine. She served as an observer intern at the All India Institute of Medical Sciences, AIMS Delhi, where she witnessed post mortem proceedings on cadavers, gaining rare, first hand insight into the final stages of death investigation. Her research work is equally impressive, focusing on the detection of organophosphorus compounds using advanced chromatographic techniques like HPTLC. She has also been trained in biosensing technologies to address antibiotic abuse, showcasing her commitment to public health safety. From animal handling to analyzing poisons in the lab, she covers the full toxicological spectrum. Please welcome Ms. Jyoti Chauhan. Finally, to connect the science to the procedure, we have Ms. Avishikta Sinha. Ms. Avishikta is a scholar from Chandigarh University with a specialized focus on the medico legal aspects of forensic science. Her practical experience is deeply rooted in the institutional framework of investigation. She completed critical internship at Bakura Sammilani Medical College in Department of Forensic Medicine and Toxicology. There, she was trained in crucial procedures of post-mortem report writing and the preservation of viscera, the vital organs preserved for chemical analysis in suspicious deaths. Furthermore, her time at the Bakura police station gave her the investigator's perspective, teaching her how to draft case documents and lodge FIRs, ensuring that medical findings hold up in the eyes of the law. Please join me in welcoming Ms. Avishikta Sinha. Next, I would like to invite the speakers and they would be continuing the session from now on. Over to you, speakers.
Good evening, everyone. Myself, Jyoti Chauhan, and I will be taking your toxicological session for today. Is my screen visible?
Yes, your
Yes,
screen
your
is
screen is
visible.
visible.
Okay. So, guys, can anyone tell me what is forensic science and how it is different? Like, what is forensics and what is toxicology? Anyone?
Forensic is
Forensic is
the
the
intersection of
intersection of
law and
law and
science,
science,
and
and
toxicology is
toxicology
the
is the
branch of
branch of
forensic science
forensic
where
science where
we deal
we
with
deal with
poison,
poison,
drugs, and
drugs, and
cases
cases
in it.
in it.
Yes, exactly. So, we will look at some points which will define us. 1 second, guys. So, we will look at some of the points in which we will try to understand what is forensics and what is toxicology. So, basically, forensics applies the science to medical and legal matters, and whereas toxicology is the study of poison and their effects. And the scope in forensic science is collection of the scientific evidence which can be useful in solving the cases, and while toxicology is a specialized branch which deals with the toxic substance which are being used um, in doing any intended crime or something. So what focuses in forensics is the crime investigation and justice, and in toxicology we detect the poison action and treatment. And some examples for forensics are DNA analysis, fingerprinting, ballistics, whereas for our toxicology, it is the drug toxicity, pesticide poisoning, heavy metals, and their identification. So there are some important terms which you should know, and you have already gone through these terms, like what are the branches of toxicology: clinical toxicology, environmental toxicology, occupational, analytical, and genetic. So, and then the principle of toxicology, where we study about dose and response, like what amount of dose will be given and how the response will affect the person. And the hazard and exposure. Hazard and exposure will define in terms of environmental, like if there is like there is if high amount of carbon dioxide poisoning, that exposure will lead to some hazards, like death or damage to the body. And individual sensitivity defines how the person responds to that toxic substance. In the next, we have route of exposure. In toxicology, there are several routes by which the toxins can be administered, like oral, through pills, drinking water, anything; inhalation in form of gases; dermal by um, any cream; and injection, like IV, IP. These are the things. Then we have type of poisoning: acute, subchronic, and chronic. Acute poisoning is when we are administering a, a high dose of toxic substance in short duration of time. So in all, we can determine the acute toxicity within 24 hours, whereas in subchronic, the dosage is um, low as compared to acute, and duration is high. Like we can identify the subchronic toxicity in 1 to 3 months, and for chronic, it is basically prolonged exposure of toxins in very small amount. And For the chronic toxicity exposure, we will have a case study also. Now, the nature of poisoning, like homicidal, suicidal, accidental, occupational. Homicidal given by a person to kill someone, suicidal, uh, a person committing suicide or killing or taking their life by themselves. Accidental, like there are many cases जिसमें बोलते हैं कि बच्चे ने फिनाइल पी लिया गलती से, like that cases. And occupational are basically linked with the your workplace. Like there are many workplaces जहाँ पे फाइबर्स का काम होता है, ग्लास फाइबर्स का काम होता है, जिसकी वजह से जो वो फाइबर्स होते हैं, जो वर्कर्स होते हैं, उनकी लंग में जाकर के टॉक्सिसिटी इफेक्ट कर देते हैं, यानी कि उन्हें चोक कर देते हैं बेसिकली. एंड the main and the most important thing is based on the origin. So poison is basically defined or divided into two parts: biological and non-biological. For biological, we have plants and animals. In plants, mushroom poisoning, tongue nut, castor bean, foxglove. And for animal, we have insects, snakes, lizard, fish. And for non-biological, we have naturally occurring like gases, volatile substance, non-volatile substance, and man-made things, and non-biological things, which are agriculture, industrial, air pollutants, and drugs. These are the important terms and things that one should know while studying the toxicology. And we will now understand how, as a toxicologist, forensic toxicologist, we will determine the poisoning cases or any other cases which need toxicological aspects. Firstly, the suspicion of poison. Basically, we don't do postmortems. We only do these things when we are asked to. Like कोई बहुत emergency case है जिसमें फैमिली को डाउट है कि ये सिर्फ डेथ नहीं हो सकती है, इसके पीछे और भी कोई रीजंस हो सकते हैं. तो इन दैट केस medico-legal forms are filled and then toxicological processes are done. Then we have evolution of circumstantial evidence, like जो forensic फील्ड पे जो forensic साइंटिस्ट जाते हैं वो कुछ एविडेंस कलेक्ट करते हैं. लाइक किसी ने सुसाइड किया है तो वो बॉटल्स कलेक्ट करेंगे, किसी का मर्डर हुआ है तो वो नाइफ या कुछ ऐसी चीज़ें कलेक्ट करेंगे जिससे हमें पता चल सके कि क्या क्राइम हुआ है. एंड इन केस ऑफ पॉइजनिंग हम जो विक्टिम है उसके घर वालों से बात करेंगे, लाइक कैसा बिहेवियर रहा है और कौन कौन सी मेडिकेशंस जो पर्सन है वो ले रहा था, ये सब चीज़ें. देन वी विल डू दी clinical features like antemortem, postmortem. Then we have postmortem examination, external and internal finding. Then we will collect the samples, viscera samples, blood, urine, gastric content, viscera, hair, nails, bones. Then proper preservation and sealing of those samples. Then the preliminary test. प्रीलिमिनरी टेस्ट आर दोज टेस्ट जिससे हमें बस पता चल सके कि ये चीज़ एक्चुअली में हुई है. लाइक देयर इज़ अ क्राइम सीन जहाँ पे रेड स्पॉट्स हमें दिखे हैं, बट क्या वो रेड स्पॉट्स ब्लड ही हैं या नहीं, उसे चेक करने के लिए वी डू प्रीलिमिनरी स्क्रीनिंग टेस्ट. एंड आफ्टर दैट वी डू द कन्फर्मेटरी टेस्ट कि अगर हमें पता चल गया है कि हाँ ये ब्लड ही है, नाउ वी विल डू कन्फर्मेटरी टेस्ट कि क्या ये ब्लड इंसान का है, एनिमल का है और किसका है. Then we have the forensic opinion कि cause of death क्या है, role of poison क्या हुआ था. And in this manner we will define कि ये homicidal था भी नहीं, suicidal था भी नहीं, या accidental था. And उसके बाद medico legal reports बनी जाती हैं और courts के पास जाती हैं जहाँ पे proper उसका दिखाया जाता है कि हाँ कि जो चीज़ हुई है वो एक्चुअल में हुई है, इसी तरीके से हुई है. नाउ द फॉर डायग्नोसिस ऑफ द पॉइजनिंग, जैसे मैंने पहले बोला था केस स्टडी की जाती है जिसमें हम देखते हैं कि जो पर्सन है क्या उसकी डेथ सडन हुई है या कोई इलनेस थी या फिर उसकी लाइफ में कोई उसे थ्रेट दे रहा था, या उसकी किसी से लड़ाई हो रही थी, या वो डिप्रेस था, फाइनेंशियल प्रॉब्लम्स थी। और फिर हम उसकी हिस्ट्री देखेंगे, जैसे कि वो कौन सी मेडिसिन लेता था, या अल्कोहल कंज्यूम करता था, या फिर किसी वो ऐसी फैक्ट्री में काम करता था जहाँ पे बहुत ज़्यादा ऑक्यूपेशनल एक्सपोजर हो। एंड सीन फाइंडिंग में हम बोल सकते हैं लाइक एम्प्टी बॉटल्स, टैबलेट, पेस्टिसाइड्स, दीज ऑल थिंग्स विल बी कलेक्टेड बाय द फोरेंसिक साइंटिस्ट which will go at the scene of crime. Then we have the clinical features, antemortem यानी कि पोस्टमार्टम से पहले, यानी कि एक्सटर्नल फाइंडिंग्स बेसिकली, कि उसमें क्या साइंस एंड सिम्टम्स आए थे। एंड दीज आर सम ऑफ द क्लिनिकल फाइंडिंग्स, लाइक स्किन कलर में क्या चेंजेस आए थे। अगर वो चेरी पिंक था तो कार्बन मोनोऑक्साइड की वजह से उस इंसान की डेथ हुई थी। फ्लैश पिंक स्किन आया था, यानी कि अल्कोहल, कोकेन, साइनाइड इन सब से हुई थी। एंड स्किन चेंजेस, यानी कि स्किन में भी जो जो चेंजेस आते हैं, प्यूपिल डायलेट है या कॉन्स्ट्रिक्टेड है, ये सब भी देखा जाता है। एंड हियर आर सम अदर एग्जांपल्स, लाइक इंसान को, जैसे कि अगर कोई लाइव पर्सन है, मतलब जिंदा पर्सन है, उसको अपने चीज़ें देखनी है, तो वो भी इसके थ्रू हम डिटेक्ट कर सकते हैं, यानी कि उसे किस चीज़ से पॉइजनिंग हुई होगी। Now the general aspects of postmortem in forensic toxicology. इसमें हमें देखना होता है कि establish the cause of death, manner of death, time since death, to establish the role of xenobiotic in the death. xenobiotic भी हमारी मतलब जो डेथ होती है, नेचुरल डेथ या जैसी भी डेथ होती है, उसमें xenobiotics का भी बहुत इम्पोर्टेंट रोल माना जाता है। एंड जो केसेस होते हैं, अगर मतलब जैसे कि वो लीगल केस होंगे, तो वो किस हिसाब से, मतलब कि उसमें क्या क्या ऐसे चीज़ें होनी चाहिए जो लीगल एस्पेक्ट्स में आएंगी, जैसे कि कोई वायलेंट या क्राइम या सुसाइडल एक्सीडेंट्स हो गए, या फिर किसी हेल्दी पर्सन की अचानक से डेथ हो गई, कोई रीज़न पता ही नहीं चला। इन सब चीज़ों में एक मेडिको लीगल चीज़ देखी जाती है, और ये प्रॉपर ज्यूरिसडिक्शन और मेडिसिन के अंडर आता है। देन वी हैव द कलेक्शन ऑफ सैंपल्स, जैसे मैंने बोला था कि जो सस्पेक्टेड जो हमारे सस्पेक्ट्स हैं, कि मतलब विक्टिम्स हैं, कि हमें मतलब हमें जानना है कि उन्हें किस तरीके की पॉइजनिंग हुई है, तो हम उनके सैंपल्स लेते हैं, जैसे कि ब्लड लेते हैं। ब्लड से हमें क्या पता चलता है, अल्कोहल, ड्रग्स, साइनाइड एंड कार्बन मोनोऑक्साइड। कितना अमाउंट लेते हैं, 10 to 20 ml लेते हैं, एंड प्रिजर्व करते हैं सोडियम फ्लोराइड में। ये होता है हमारा पेरिफेरल रेफरल के लिए। और हम लेते हैं ब्लड सेंट्रल से भी। फिर हम यूरिन कलेक्ट करते हैं, गैस्ट्रिक कंटेंट कलेक्ट करते हैं, एंड मोस्ट इम्पॉर्टेंट हम कलेक्ट करते हैं लिवर, किडनी, स्प्लीन। लिवर, किडनी, स्प्लीन एक मेजर रोल प्ले
Excuse
करते
me,
हैं।
ma'am.
यस,
I'm really sorry to interrupt. Can you please use English? Because I can see in the chat box some students are actually requesting you to use English, as some have joined from South India and are not very familiar in Hindi.
okay.
Okay, thank you so much.
Okay. So the important things which need to be collected are the viscera samples, and the viscera samples are liver, kidney, spleen. These are the things, and they play a major role, because like when we consume any drug or any medicine, it directly goes to the liver, where we see the first past metabolism. And liver stores, like for paracetamol, liver stores it up to 6 months. So we basically take liver, kidney and spleen. And for liver, we take the right lobe, and it should weigh around 300 to 500 grams. And for kidney, we take a whole kidney, because in case we are not able to detect any poison in the urine, but we will be able to find some traces in the kidney. And some other samples also, like hair, nails, bone marrow, soil. For nails, like this sort of between two peoples, one has murdered the other. So while struggling, he or she may have scratched the accused. So through by nail clipping, we can collect the DNA sample for the accused and give justice to the victim. And in case, like the case hasn't been solved, so the body ultimately get cremated. So in case of that, and after some time, we get to know that this was the poisoning or this was the case, but it haven't been solved. So we can collect some bone samples and bone marrow and soil. For soil, like there are some poison like arsenic, cyanide. They um, they not only affect the person, but if the person is being burrowed uh, burrowed, so the cyanide mixes with the soil. So these are the things which needed to be collected for the detection of any kind of poison. Now the toxicological analysis. In this, we have two types of analysis: preliminary and confirmatory. For, as I have told you, like preliminary is the test where we check whether the thing is what we are thinking is there or not. And for confirmatory, we do the final test, which will be leading and will be wrote down within the medical legal forms and will be presented in front of the court. So uh, uh, for definition, for preliminary is the initial test to suspect presence or class of poison, and for confirmatory defines the test to identify and quantify poison. And as you can see, it is not that much difficult to understand. So these are the toxicological analysis. Sorry. Now this is the toxicological analysis request form, which will be requested and filled by the family of victim and the chain of custody when there is any legal proceeding regarding the collection, identification, or post-mortem, and or the uh, items which will be handed over to us. It will go through a chain of custody where we will uh, provide the information in a sealed pack. It will be sealed and passed to the other, so that there is no changes or mishappening between them, like they don't get merged like this. Now the case study. This is very interesting case study of Lady Lafarge and Aquatofana. It is a classical case study. Um, in this, there was a lady, lady named Mary Lafarge. She was not happy with her husband, so there was another lady, Tofana. She used to sell poisons in forms of cosmetics, perfumes, like this. So she used to sell this for the person who are not happy with their marriage and husband and wife for them. So the Lady Lafarge bought that Aquatofana. The bottle says Aquatofana, and she used to mix it with the food and serve it to her husband. And after certain period of time, her husband died. and no one suspected that it was a murder, not accidental or something. So after sorry, so initial test, local doctors tested, and they came negative. Like, there is nothing to be suspicious about this. It is totally a natural death. But there was Matthew Orfila, who performed the Marsh test, and a Marsh test, it was used to identify the arsenic. So by this test, it was confirmed that the Lady Lafarge has poisoned her husband, and by this he has died. So, and using the Marsh test, which was after that, Matthew Orfila was defined as the father of modern toxicology. So he collected some soil samples, tested it, and came positive that he was given arsenic. So this was the first case. Now the second case, you may all have heard about this Korean cyanide, Jolly Joseph case, in which she killed her 6 family members by giving them cyanide. Like, it, it is till now a very famous case. You, she was born and brought up in a very middle class or low class family. She was very fond of expensive things and luxurious life. So when she met her husband, she was very relaxed that she don't have to work anymore and she will be living her life very peacefully. But after some time, her mother-in-law asked her to start doing some job, and um she used to earn something. So she got very upset and she started giving her mother-in-law cyanide. So the first murder was her mother-in-law, second was her father-in-law, third was third was her husband. And when her husband died, his uncle suspected that there is something very unusual about this death because his body was turning blue. So he questioned and asked for medical legal procedures. And Jolly Joseph, she got angry and she ultimately poisoned her uncle also. And she met, and um, and after that her uncle also died. They were her neighbors. They were husband, wife, and a kid. She used to like that lady's husband, so she killed that man's wife and daughter. So ultimately, she killed a lot of person to be in a luxury lifestyle. So modus operandi was Jolly Joseph illegally poisoned her victims with cyanide laced food and water to gain control of the family property and finances. The victim include her mother-in-law, father-in-law, first husband, her husband's uncle, her current husband's former wife, and their 2-year-old daughter. Now, what were the forensic toxicological evidence? Exhumation of the bodies conducted year after burial, and bone marrow, viscera, remnant, soil around the remains were collected. Then cyanide detected despite decomposition due to chemical stability of cyanide residue. It was not able to, the test came to be false, like there is no trace of cyanide. Then the case was reopened in 2019 after the complaint from the surviving family members that lead to exhumation of the bodies and forensic analysis that confirmed cyanide poisoning. Jolly Joseph was arrested and reported confessed to the crimes. So after many questioning and proceedings, she finally accepted her um, crime. So these are the 2 case studies which are very famous and classics. Thank you everyone. Any questions?
It's okay, ma'am. I think let's keep the questions for the end. Okay, so at the end we will be having the Q&A session. So now I would like to request Miss Abhishekta.
Yes, can you please share the PPT, please?
Yes, just give me a minute.
Okay, okay.
Can anyone please let me know if my screen is visible?
No, it's not visible till now.
Just a second. What about now?
Yeah, it's showing now.
Yes, sir.
It's visible, right?
Yes.
Okay, just give me a minute. I'm sharing the slides now. I will try to open the slideshow mode. So in case the slides do not change or there is any kind of issue, please let me know. Accordingly, I will again share my screen if required. Done? Am I audible?
Yes, you are audible, sir.
Okay. So can you see my screen now?
Yes,
Yeah.
sir.
Okay, done.
So, good evening. Today, my topic is a very medicine, forensic medicine. So, I will just give a big description about an overall view of forensic medicine, and rest of the details my co-partner will Priya will describe you all guys. So, let's start with forensic medicine. So, what is forensic medicine? Is anyone can tell me? Guys, can you please make it an interactive session on both the end? It's not looking good that only I am speaking. Is anyone? Okay. So I will say the forensic medicine is nothing. It's just basically a branch of forensic only, which basically deals with the application of medical knowledges for the purpose of law and the administrative justice. Can you please move to the next slide, sir? Okay, so there are some branches of medicine which is basically used for the legal purposes, for the legal aspects. Like there is can say forensic pathology, or you can say forensic toxicology and forensic psychiatry. So for forensic pathology, is basically aim of the pathology is to identify the cause and manner of the death by doing the detailed autopsy of any body. And for forensic toxicology, you can say that to analyze the drug substance or the chemical substance which is basically found in that particular body or in that particular postmortem. So basically we use viscera samples for collecting as my previous co-partner have already described what kind of symptoms we can see for the toxic substance effects. So basically this is for the forensic toxicology. And forensic psychiatry is nothing, just they give the mental state if some patients or the some kind of victim or some kind of person is involved in any matter of legal or illegal things, like illegal things or any drug, they're taking excessive amount of drugs or any illegal things. So for giving them a mental assessment, so forensic psychiatry is used. Next slide, please. Hello, sir. Can you please move to the next slide? Hello. Yes. No. Before there are slides also. No. Okay, importance. Huh? So, what are the importance we can say about the forensic medicine? Is can it's provide as the forensic medicine reports or the autopsy reports plays a very vital role in the courtroom for giving a proper overview of that person. Like what are the symptoms? Like what are the body condition? What during the postmortem? It also helps to ensure the medical experts to give justice, and even it also helps to sometimes it helps to reconstruction also, and also to establish the facts in the crime scene. Next, please. Okay. So
Is it
now there is a role of what are the role in crime investigation for the forensic medicine. So as there are some medical clues, like as previously she said, my co-partner said, like there are some chemical founds in that person's body. To analyze that, to medical analysis clues helps a lot in setting a crime investigation, like support identification and reconstruction of events. And the basic area where the forensic medicine focuses during all the total crime investigations and during its procedure, like what are the injuries mark, like if there is any injury mark, they even that notice that came in the report. Even the death investigation, like it either poisoning or by alcohol or by any drug effect. Like even the medical examination is also done, and interpretation of medical evidences, like the reports they're making. Next slide, please. Okay, so I will over this to Priya.
Swapnil, can you stop the screen sharing so that I can share? Is my screen is visible?
No, ma'am.
No, ma'am.
Okay.
No, ma'am.
No,
Is
ma'am.
it visible now?
No.
Okay, just give me a minute. Screen is visible now?
Yes, ma'am.
Anybody?
Yes, ma'am.
Thank you.
Yes, ma'am.
So, good evening, everyone. This is Priya this side. So, let's see the today's tidbit. So, Avishikta has given the basic introduction of forensic medicine. So that things I will be covering today is post-mortem changes, types of injury, autopsy, forensic biotechnology, DNA profiling, forensic serology, biological evidence handling will be taken by Avishikta, and application is will be taken by me. So before going to the topic, do anybody know what is called thanatology? Let's be for you guys can be attentive so that the topic we are going to see will be more interesting. So anybody from the side can.
The participants are requested to please unmute when the speaker is talking to you. Please make this session interactive because I know, guys, that you all have some idea. Your answers need not always be correct, but at least interact.
Okay, let it be. So before going to the topic, we will see what does the thanatology means. Thanatology basically deals. It's called thanatology means it's called the study of death. Okay, so it is one of the specialized branch of forensic medicine which deals with death and early changes of death, late changes of death, and everything which is occurring in human body. So Thanos in the word Thanatology, Thanos means death. Okay, so we will be seeing what the changes happens after the death. So death is classified as two main branches. So one will be somatic death and molecular death, and another one death is also there. It's called brain death. So in somatic death, the irreversible will be stoppage. That means the you can see in this picture. So the functioning of heart, brain, and lung as well, everything will be stopped. Yeah, and this you can, and like in this the stoppage of the thing will be in short time, like the function of heart, brain, and lung. The function of this will be short time, like in movies or in anything you are seeing. A person will be declared that he is dead, and after some time they will come back to life, right? So you guys might have seen those kind of thing in movies or real time also. They will you can all have heard about this. So this term is called somatic death. So this will be not monitored by the any medical equipment. So the this stoppage of heart, lungs, and brain function will be in short time. It will be. And next one is molecular death. In this molecular death, the cells of an individual will be die gradually. So this occurs 2 to 3 hours after the death, somatic death. Okay, so in brain death, complete and irreversible loss of brain activity. The functioning of the brain will be totally stopped. So these are the signs of the death. So there are three main signs after the death that are called as algar mortis, lever mortis, and rigor mortis. Uh, while these three, cessation of respiration, pupil changes, and cessation of circulation are the signs that are seen right after the death. So basically you guys have seen that if somebody is they are going to their pulse is low, they will check their eyelids. Either the pupil, if it's not moving, they will declare that the person is dead. And if they are not having uh, pulse, or if they are not having the breathe, they will declare them as dead. So signs after that, there are immediate signs, early signs, and late signs after the death happens. So the early signs will be absence of pulse and respiration, loss of reflex, fixed and dilated pupils. And early signs will be algar mortis, liver mortis, and rigor mortis. And the late changes will be putrefaction, adipose formation, mummification, and skeletonization. So let's see what is called algar mortis. Algar mortis is called the cooling of body. So literally, basically, if a person is died, uh, their body temperature gradually it start to decrease. Right. Just give me a minute. So in which the ratio of surface area of body masses is large, cool more quickly. These are several formulas for estimating the rate of postmortem cooling. Like when the person is declared dead, this process only starts because it's the early stage. You might have seen that their body starts to cool. So this cooling will be in the 0.5 to 1 degree Celsius per hour. So this is the time estimation. So per hour, this amount of body temperature will be gradually decrease in normal condition. So there are other condition like in winter and in summer, the body condition will be normally decrease. So this body condition before antemortem, that is before postmortem, the body temperature actually varies from 93.74 degrees to 100 degree Fahrenheit. Approximately 1 hour after the death, the body cools at the rate of 0.78 degrees Celsius, that is 1.4 degree Fahrenheit per hour. After the first 12 hours, the body loses about 0.39 degree Celsius, that is 0.7 degree Fahrenheit per hour, until the body reaches the same temperature of the surrounding. Time of death determined by the body temperature calculation is always expressed as a range of time because it cannot be calculated exactly. So uh, there is a thing that there is a formula that is calculated for the temperature, that is the time since death is equal to temperature of the body recorded after the death by the rate of fall of temperature. So the medical officer or anybody who will be seeing the postmortem, they will calculate this to get the algar mortis value. So the temperature, the person who died, so at that time they will calculate the temperature, and they will divide it by the rate of fall of temperature, that is the surrounding. They will calculate and they will get the algar mortis. So this is the things you can see, these are the things happen after the death. So you can see these things. So liver mortis, this is it is also called as postmortem lividity, liver mortis, or postmortem hypostasis. Discoloration that appears on dependent position of the body other than parts exposed to the pressure after the heart ceases to beat. So algar mortis, we have seen that it's the fall of temperature, that is cooling of body. In this liver mortis, the coloration of the body is seen, like you can see in this. This is the discoloration of the body. It's seen after the death. Usually appears 30 minutes to 2 hours after the death. it will be well developed like after 6 to 8 hours after the death. And it after it's well developed, once it reaches 8 hours, the color will be fixed as you see in this, right? You can see that in some area the color is fixed, and in some area it is not there. You may ask me why it's so. So this might be the pressure of the any clothes or any other external things. So the you can see these are the marks. So distribution, it depends on the body position, absent in pressure area. Like you can see in this pressure area might be there garments or any other thing, so that it's not, the coloration is not visible because the garment give a pressure to our body. So in here and all you can see that coloration is happen. So the color variation will be bluish purple. Bluish purple color variation means it's normal. Cherry red means it's carbon monoxide poisoning. Chocolate brown means it's methemoglobin. So forensic importance of this is indicates the position of the body after the death. Times this death is estimated. So this will suggest the cause of death in many ways. So let's see what is called rigor mortis. Rigor mortis develops as body's energy source like ATP is depleted. Muscle fibers request ATP for relaxation. Once depleted, actin and myosin proteins remain complex, resulting in stiffening of the muscle. Rigor mortis is to develop in all muscle simultaneously. However, it is most evident at first in smaller muscle groups such as jaw, we have seen algar mortis is fall of temperature, and liver mortis is body. So rigor mortis is saying that it's a stiffening of muscle, right? So this why it's happening, because the depletion of ATP. The ATP is synthesized in the body while the human body is working, right? So after the death, the ATP will not produce. So it is depleted. Because of this, the actin and myosin it gets fluctuated, and this causes the muscle to become rigid. So this is where it is seen is. So first the rigor mortis is seen in eyelids. So guys might have seen that when the person is declared dead, if their eyes is open, they will close the eyes, right? So this is because of the rigor mortis. If they didn't close the eyes for 1 to 2 hours, the eyes don't close because the muscles get stiffened, and then it will from eyes it will then it's seen in face and in neck and in trunk, upper limbs and lower limbs. So this is why they will close the eyes. If the jaw, you might have seen in some cases in fire injury or any other like hanging and all, if the body is stop moving suddenly, and it's not disturbed or it's not bring back to its actual position, the muscle got stiff and it will not, we can't move after that. So this will rigor mortis, it onset is onset means the time it starts, the stiffening got start is 1 to 2 hours after the death, and after 12 hours it will be fully developed. And after 24 to 36 hours it will passes off. So this you can see, this is called I can see it as this person's hand got stiff because the muscle reflex is not happening. So what are the factors which will affect the rigor mortis will be temperature, muscular activity before the death, age and physique, cause of death. So cause of death in the sense if they are died by any external or anything, like for example if they are being drowned, or if they are being hanged, if their body is moving and suddenly it stopped, the you can see in this hand like that, if the body is stopped, the muscle activity it will be stopped, and we can find that in which might be the cause of death. So forensic importance is useful for time since death estimation. This indicates whether the body was moved or not. Then let's move on to late changes of death. You can see these are the late changes: mummification, putrefaction, formation, and skeletonization. These are the late changes of the death. First, we see what is called decomposition, progressive breakdown of body tissue after death due to autolysis and putrefaction, leading to reconstitution of soft tissue. The process involved in decomposition is autolysis and putrefaction. Let's see what is called autolysis. It is self-digestion of cell by intracellular enzyme due to lack of oxygen. Putrefaction is breakdown of tissue by bacterial action, producing gases and foul smell. So, decomposition have like around five stages: the fresh stage, blot stage, active stage, advanced stage, and dry stage. So the fresh stage, no external visibles are seen. In the blotting stage, the gas formation in abdomen area is seen. In active decay, the skin slippage, blisters, and maggots are seen. Advanced decay is loss of soft tissue. Dried remains, you know that it's mummification and skeletonization. So these are the stages you can see. This is the fresh stage. The thumb finger indicates the fresh stage. The blot stage, you can see that the hand got puffed because of the gases. Active decay is this stage. You can see that maggots, like worms, it's came here. And active decay is the soft tissue gradually got decreased. Dry stages, you can see that only bone is remaining here. So the external features of decomposition is uh, greenish discoloration of abdomen, marbling of skin, foul odor, purging of fluid from nose and mouth. So purging of fluid means the fluid which usually comes out from nose and mouth. Marbling of skin will be a marble kind of pattern is seen in skin, and greenish discoloration is seen in abdomen. I will show you the pictures while we are gradually shifting to slides. Factors affecting decomposition. What are the factors will be affecting the decomposition will be temperature and humidity. So basically, if the it depends on the season, like temperature and humidity, defaultly it implies the season, right? So in summer the decomposition starts soon, and in winter it will be quite late. because of the temperature and humidity. Presence of insects, like presence of maggots or anything, this decomposition will be start. Like factors will, these are the factors affecting burial depth. So if they are burying a body or any, the decomposition starts soon. Cause of death is another factor, and body composition, the how the body adapts the environment and it composes it. These are the factors affecting decomposition. Forensic importance will be the, it will help in estimating the how long the postmortem interval has there, and assist in determining environment of death, provides clues regarding cause and manner of death. Putrefaction, decomposition of let's see what is called putrefaction. Decomposition of body tissue by bacterial action. So the first greenish discoloration of the abdomen, fecal formation of body, skin slippage, and whole order. So this is putrefaction. As you can see in the right image, you can see that greenish discoloration, and some maggots are, you can see that maggots are there, and the skin is slightly getting, you can see here, the skin is getting slippage, means it's here, this is called marbling of skin. Here also you can see this. So let's see what is called adipose formation. Conversion of body fat into waxy substance. Okay, you can see here in this, the body, it starts like, you can see it's very shiny, like some wax coating is given, and you can you can see this hand, this is called a rigor mortis. So you can see that the body of the hand of the body has got stiffened, and the legs, you can see, this is the one good example of rigor mortis, like stiffening of body. So this is adipose formation. This is early adipose formation, and this is late, like the body, it got so much adipose formation. You can see literally the bones are visible here. So this is late formation. So the condition where this adipose formation is seen is moist environment, lack of oxygen. In this cases, the adipose formation is, this preserves the body feature. Let's move on to mummification, drying and shrinking of body. This basically happen in a dry and hot environment, well ventilated environment. So the appearance is dry and leathery skin. You can see the skin being very leathery. It's not dry, it's like leathery, that the skin got sticked with the bones, and this is dry. You can see the legs of this fetus, it got literally it got dried. So in this, you can see in bone area, it's leathery. Skeletonization, complete decomposition, leave the, so we see that it's advanced stage of late changes of death. So after this, the change will stop. Time required for this skeletonization is months to year. It depends on the environment, and it depends on the individual. right? So the importance of this skeletonization is aids in identification and forensic anthropology. So it helps to identify the individual. The forensic importance of late changes is estimation of how long the post-mortem interval is have seen help in cause and of death, assist in identification of the deceased. Let's see what is injuries. So, body tissue produced by external agents such as mechanical force, thermal and on the body. Uh, so in this first type is abrasion. Abrasion is nothing would have experienced this many times as a kid or as an adult or day to day life. You, as the picture implies, when we fall on the we get more scratches, right? So it is called the abrasion. In this, the superficial loss of epidermis, that is the upper layer of the skin. So in this abrasion, epidermis is only affected, caused by friction and scraping against the rough surface. So this is caused by friction against the rough surface or by compression, such as lateral rubbing action by glance blow, a fall on rough surface by being dragged in a vehicular accident, and fingernail stones and teeth bites. So these are the cases where the lateral rubbing action. So these are the kinds of types of abrasion. Linear abrasion, you can see that will get in fingernails thrown, like small thrones in plants. If we get skin got dragged by thorn, we will get linear abrasion. And grazed or brush abrasion, this is a vehicular accident. So forensic importance of abrasion is this indicates the direction of force, and this is dragged straightly, like you can see in hand that some road or some vehicle it have got the force has got dragged. So let's see what is called the next one, contusion. So this is basically it is called bruise. So this is caused by blunt force. Okay, the rupture of blood vessels with extravasation of blood into tissue. This is happened by the extravasation of blood. Okay, so the color changes help in age estimation of injury. Forensic importance importance of bruise is this indicates the type of weapon is used and the force, because it's the contusion is blunt force injury. So we can find that which kind of weapon or tool is used, on which kind of force is used. We can identify those things, so you can see the contusion. Here clogged. Due to blunt force. So in contusion, we have seen that the blood vessel will be extravased. So in laceration, the skin tissue, it will be teared the due to blunt force. So margins are irregular, bruised and abrated. The types of laceration are split laceration, stretch laceration, avulation. Forensic importance of laceration is helps differentiate from incised wounds. So in this we can this is tearing of skin. So we can compare incised wounds, blunt force injury, and there are many kind of injuries we will see in upcoming slides. So we can identify those kind of injury by comparing these things. So this is the one of the forensic importance of laceration. You can see this is laceration. You can see that the skin got teared because of blunt force. You can see that the injury is not too sharp. For example, if any rod or any hammer got fall on the skin, or it is got somebody had beaten, because of that the layer of the tissue got teared. And in this hand you can see, because you can see that the injury is not hard, or it is not sharp. So you can identify this on seeing that it's a blunt force injury. You can see in type 1 it's like this, and type 2 and type 3. Let's see what is called incised wound. You can see that incision has happened in this area, so it got cut open, the slit open has happened. So this is caused by sharp edged weapons like hammer, sorry, not hammer, like saw, razor, knife, anything that will be sharp. This injury is caused by that which has sharp edges. Okay, so in this clean cut margins are seen, as you can see here, clean cut. You can see here, you can see that a good cut is happened here and it got incised because of that. And in this visibly you can see that here the cut has happened. So you can see the clean cut in this. So importance this indicates. So ultimately you know the incision will be happen because of the sharp weapons, right? You can see by in which direction the wound has occurred. So this, like beaten with the any knife or anything, sword or anything, if they have the force is given here only, it will get slit open like this. So you can see it's very deep here, and it's gradually getting small here. So you can say the injury, the force has given here the forensic important of stab wound. Let's see what is called chop wound. This is caused by heavy sharp weapons like axe or
Ma'am,
very
your PPT is
big
not
sword
visible.
or anything.
Priya, can you share the PPT? Your PPT sharing stop. Please share the PPT.
Is this visible now?
No, ma'am, it is still not visible.
Is my screen is visible?
No, ma'am, not yet.
No, ma'am.
Okay, just give me a minute. Is this visible? Anybody?
Yes, ma'am. Yes, ma'am, it's visible.
Yes,
Can
ma'am, it
anybody
is
say
visible now.
until where did I stop the screen got stopped? Can anybody say?
Ma'am, you are on chopped wound. Yes, ma'am.
Chop wound.
Yes, ma'am, on chopped wound.
Okay, got it. So anybody can let me know what I have already said that. So in the name itself implies that what kind of wound it will be. So anybody can just brief me what it might be. Just normal in normal terms, you can say that. Just be interactive. It's like I'm just saying. So this class is being very, very dry. So I need some interaction. Anybody? Okay, let it be. So as I said, the chop wound is caused by heavy sharp weapons. So it might be a axe or it might be a big sword or anything. So this is happen. You can see the head of the. You can see that here the head itself got chopped. So and the hand got chopped here and the back portion after here in the trunk it got chopped. So you can see that it got charred because of very heavy weapons only. So in this, features like incision and laceration is seen. Incision you can see here, and the laceration, the skin is got wounded, so you can see laceration here, as well as incision here. So in this, both features is seen. This is often associated with fractures. The forensic importance indicate the how heavy sharp weapon is used. Okay, this is the forensic importance of this. So let's see. Now we have seen this long was flesh injury, like these are the mechanical injury. So let's see thermal injury. This how the injury is happened because of heat or cold environment. So these are the kinds of burns which is seen in heat injury, like thermal, because of burns it happened, hot burns. So this is first degree burn, second degree burn, you can see the blisters, you can see the third degree burns, and in here you can see the cold burns, like cold injury. So what we'll see, what is called burns. Burns is caused by dry heat, flames, chemicals, electricity, etc. By these factors, burns is happen. So there is 3 degree of burns. So as we see, a first degree will be redness, and the second degree will be blistered, will be formed, and the third degree we might see that it will be blackish color and or bluish purple color. So in third degree, charring of skin is happened. It means the deconstruction of skin is happened. So the forensic importance of the burns is we can find the cause of death and manner of death. So see, in injury, the most important forensic important in injury is we will be able to find the cause and manner of the death. So this is the one of the most forensic important we will be seeing in all injuries. Okay. So this is the 9S rule where we can see how the burns is calculated. So if you add these things, you will get uh, around how the bodies get burned fully. So front and back of the body, like head, the total head, we can see the trunk or everything. This is calculated. So this is how the body is like the burn is calculated. You can see here. If you add this, we will get the 1 minute. So let's see what is called scandals. This is caused by moist heat. So my moist heat in the sense, if the person is affected by heat water, or if somebody had poured hot water on them, or if it had happened because of the steam, so this scandals will happen. This produce blisters without charring. blisters in the sense we have seen. Here we are seeing now. So in this chair this happened. So in that scandal only blister will be formed, like only this will be formed. Here it's charring is happening. You can see charring means peeling of skin. So this is happening. So in scandal only blister will be formed. You can see here this is called blister, like a bump of skin which happens because of heat. You might have experienced this when we are. So cold injuries. This is basically frostbite and hypothermia. Tissue damage is happened due to extreme cold. So forensic importance is exposure of death is seen here. Then the chemical injury. This is called by this is caused by acids and alkalines. Uh, you can, as my co-partner has said, that this will be help in finding the which kind of chemical or which kind of force any agents is used for doing the crime. Electric injuries, this is caused by the current or electric force which is given in the passed in the body. Entry and exit wound may be present. Joel burn seen at the contact site, in the sense if somebody have got the heating rod which we used in heating the water, right? If they are holding that rod and they got current, electric current, because of that we can see the contact where the uh, rod got contact in hand, and we can find the contact side, and we can estimate that here might the injury might be happen. Radiation injury is caused by the exposure of radiation. Damage to skin, bone marrow, and organs are seen in this case. This kind of injury is rare, but it is very significant in the case of industrial accidents. Firearm injury. So this is the one of the important injury in forensic aspect. This is caused by for firearms like bullets, pellets, or any other firearms. So the entry wound will be very small in this case, and the exit wound will be very big, because the pellet or any bullet or anything, it will be exit by the entry exit wound, right? So the wound will be big. In this case, blackening, tattooing, and burning is seen in this kind of injury. Forensic important of this is we can identify the range of fire and the weapon is used, because if we can identify the which kind of pellet is used, or which kind of bullet or anything is used, and we can identify what kind of firing weapon has been used. You can see these are the exit wound because it's big. This is tattooing. Blackening is this is tattooing. Like you can see the mark of the smoke released from the firearm. This is blackening. So this is the firearm injury. You can see here entry wound. This is exit wound. Because the exit wound is bigger and it got a more deconstructed the skin. Medical legal importance of injury is as it determine the cause and manner of death, as I said before. It helps in identifying the weapon, which kind of weapon is used. For example, in blunt force injury, we can find that if blunt force is given, is applied in incision, and in chopped wounds we can identify which kind of sharp weapons is used. So we can ultimately we can find which kind of weapon is used. This will be very helpful in constructing the crime scene, like reconstruction. This also help in estimating the age of injury. So let's see what is autopsy. Can anybody say that what you guys have understood what is called autopsy? Anyone?
Study of dead bodies.
Okay. Yeah, it's
Study of
good.
the body by which the death has happened.
Okay.
Study of dead body to find the cause of death.
Yeah. Great. Thank you guys for answering. As you guys said that postmortem, like autopsy is a post-mortem examination. uh, these all things we have studied: injuries, early changes, late changes. These will be studied while doing the post-mortem. If any injury is seen in body while doing post-mortem, they will do the record while doing the autopsy. So this is the post-mortem medical examination of body performed to determine the cause of death, manner of death, time of death, and document the injury, disease, and any poisoning is have done it to the person. We can study that things. So this is basically study of a body after death, which we can find the cause, manner, time, or any other external things have happened to the body. So it is the one of very important in the case of medico legal to provide scientific evidence for the court. So purpose of autopsy, why we do the autopsy? As we see it, we need to find the cause and manner of death, detect the injury, disease, and poisoning, collect evidence for investigation, confirm identity when it's unknown. Okay, so to support medico legal reporting and in court testimony, autopsy plays a major role. There is types of autopsy: medico legal autopsy, clinical or pathological autopsy, physiological autopsy. So medical legal autopsy, this is involved in recurring in unnatural, suspicious, or sudden death. In this case, these autopsies are done. Clinical or pathological autopsy is done to study disease for medical learning. For example, COVID or any other unknown disease is affected to a person. This kind of autopsy is done to study the how the disease or any other thing is caused. It is basically used to study the disease or any other external factors which is unknown. Like unknown in the sense it is new to the medical field. So psychological autopsy is to evaluate the mental state in suspected suicide. So this is psychological autopsy is done in this case. Basic steps in autopsy. Let's see what are the steps. First is external identification. So this external examination, this will be identification of the body. Photograph will be taken and clothing checks will be done. Height, weight, scars, tattoos, decomposition signs are noted, and the injuries, ligature marks, gunshot wounds, burns, etc. is seen, like they are examined. uh, Collect the trace evidence like hair, fibers, bullet, soil, anything is seen. These are collected and they are documented in the external examination. In the internal examination, a Y-shaped incision is done in thoracic and abdomen region. You can see here Y-shaped incision. See here Y-shape. The incision will be start from here, and they will put a Y shape and they will open like this. So Y-shaped incision is done in abdomen and thoracic region. In this, the examination of heart, lungs, liver, brain, etc. is done. They are taken, and the small part of this, like total organ is weighted, and the small part is cut for the further examination. Look for internal hemorrhage, fractures, disease, poison signs, etc. These are seen in internal examination. So external examination, you can see any bruises are there, gunshot wound, ligature marks, tattoo, decomposition. These are seen in external examination. In internal examination, these things are examined and samples are taken for the toxicological examination. So evidence collection, blood, urine, vitreous humor, stomach contents are taken. Tissue for toxicological histopathology, DNA. For these things, the tissues are collected. So basically, if the wound is there, they will measure it using the scale, and organs as well. You can see it is weighed. Firstly, it is weighed, and they will see the length and breadth of it, and they will document it. You can see here. So these are the kind of incision made in autopsy. This is Y shape. You can see. This is this kind of incision are made in autopsy. So documentation and outcome of autopsy. Notes, diagram, photograph, labeled of sampling is done. Findings written in post-mortem report. So in this post-mortem reports, they will write what are the findings they have seen. For example, if they are identifying any tattoo or any kind of injury or any kind of suspected things, they will be noted. And what are the moles, uh, any anything which is seen in the body, it's which is unusual, it is noted in written form. Outcome will be cause of death, like hemorrhage, myocardial infractions, asphyxia, and manner of death. They will be seen like natural death. It is done by asphyxia or hanging or poisoning. Manner of death will be also seen. So this is how an autopsy report is written, and this is how basically the diagram is hand drawn by the medical officer. So they will be drawing the body as hand drawing, and they will be, you can see here, they will be mentioning what are the things they are seeing. They will be labeling it. So this is how, these are the things which are seen inside the body. So you can see that brain, it is, they have mentioned the weight of the brain. Everything will be mentioned in this. Let's see manner of death and asphyxia. These are the manners how the death has been occurred and they are seen. So manner of death, there are these accidental, natural, suicidal, homicidal, undetermined. These are the manner of death. Asphyxia. So let's see what is called asphyxia. Asphyxia is a cause of death that may be fall under suicidal, homicidal, or accidental manner depending on the circumstances. Anybody have anybody heard or anybody know that what is called asphyxia?
No, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay, let it
When
be.
body get less oxygen, then the due to that it
Yes,
just
ma'am.
Mm, yeah. So
like when the body don't get sufficient oxygen, so
Oxygen.
then the person get died. Then that condition, in that condition, it is termed as asphyxia or death.
yeah, as the participant is implied, so these are the kind of asphyxia, these are the examples for asphyxial death where the body fails to get the oxygen, and in this stage the individual dies. So these are hanging is one of the time example for asphyxia, ligature strangulation, smothering, drowning. These are the kind of asphyxial death which is seen. So basically, asphyxia is the term where the body fails to get oxygen or lack of oxygen. The individual tends to die. So this is called asphyxia. So in mechanical asphyxia, let's see hanging. So physical abstraction or pressure affecting breathing. So body is suspended by a ligature or on the neck. Weight of the body tightens the noose. Noose in the sense, wait, let me show another picture. Noose in the sense ear. This area, the ear, this is called noose. This is ligature.
And nose means not.
Yeah. Here the knot. I'm saying this.
Not. Okay.
So this is the, as the participant said, this knot is noose. This is ligature. So in the hanging, when the body goes into ligature, because of the body's weight, the noose get tightened and the death is happened. So weight of the body tightens the noose. So in this case, the fracture of hyoid bone is seen. You can see hyoid bone in the sense here. In this area, the fracture is seen here. C6 and C7 region. Let's see what is called strangulation. So you guys are cleared with asphyxia, right?
Ma'am.
Yes.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
So in asphyxia, first one we have seen the hanging. Second one is strangulation. This is the force that is externally applied to the neck region. So this is called strangulation. Like somebody is forcing your neck, and this and you are getting absence of oxygen. You couldn't get the oxygen, and you tend to die. So it is one of the example of strangulation. So that strangulation which I said now is called throttling. in where a manual strangulation using hands or fingers is done in this. Key features, the key feature we can see in throttling is we can see nail marks in the throat, bruising on the neck, hyoid bone fracture will be possible. So these are the features which we seen in throttling. Ligature strangulation is ligature we have seen it, as I said in hanging, it is that rope which comes near the neck, so that is called ligature. If somebody have done strangulation using the ligature, for example, if somebody is putting eye card or belt or any shawl to tight your throat, so it is called ligature strangulation. um, Ligature tightened without suspension, like the ligature is tightened without any gap. So yeah, the key feature seen is horizontal mark below the thyroid cartilage. Mugging, it is the using forearm or elbow around the neck. So this is mugging is I will show the picture for you for better understanding. Um, let's first see what are the features, and we will see the picture. It will be quite understanding. So in mugging, mugging is basically seen in assaults like sexual assault, child assault, any other assault. Mugging is seen there. Bandsdola is a stick placed behind neck or rope and pulled. So in this is called bandola. Uh, this is seen in rural cases in India. Garroting. This is sudden tightening with a rod or stick. This is seen in homicidal. So you can see throttling. As I said, somebody is tightening your neck with hand. This is ligature strangulation. Either this rope can be used or anything which is doing strangulation for us. This is mugging. You can see forearm is used here. And bandsdola is a stick or any bamboo that is placed here. And this is garroting. Wire or cord is used here. So these are the signs seen in strangulation. Like cessation is happen in eyes. You can see the blood vessels got cessated. Like you can see the blood vessels here drooling, and you can see the fingerprints, ligature mark. These are the signs and symptoms of strangulation. So let's see the application of this. Establish the cause of death, identify medical, traumatic, or toxicological region for death, determining the manner of death. In crime investigation, it supports, like, is it caused by any injuries, or is it what kind of crime it's happened, like homicidal, suicidal, or any other crime. It is used, it is help in crime investigation. Verification of medical treatment, in insurance and legal purposes it is applied. Public health and research also it is used. So drowning, as the picture implies, when the person when the fluid enters to air passage, preventing the gas exchange, this term is called drowning. So fresh and salt water drowning have will be happen. So diatom identification helps in finding the clues. So let's see what is a diatom. Diatoms are unicellular phototropes that evolved some around 1,808 million years ago. So this is one of the eukaryotic algae. Oxygen synthesis on Earth is one of the most important source of biomass in ocean or fresh water. So this diatom cells contain a silica wall, which helps in identifying the um atmospheric oxygen. So this is widely distributed among species. So it can be help in identifying the species variation by location, because uh, it is available. The species will be more. So we can find the species and variation according to the location. They are resistant to acid because it has silica composition, and it plays an important role in forensic investigation of drowning cases. The role of diatom in drowning. Let's see what its role in drowning cases. Diatom enters the lungs and with aspirated water during antemortem drowning. From the lungs they enter the bloodstream, travel to circulation, and get deposited in distant organ, especially in bone of femur and sternum. Presence of diatom in internal organs confirms water inhalation while alive, helping to distinguish true drowning from postmortem immersions. Matching the diatom species in the body with those found in suspected drowning water also established in the scene of drowning. So, basically, if any a person is drowned, the fluid is seen in their body. The fluid is taken for diatom test to confirm the presence of diatom in the body. So in this detection of diatom in tissues, so basically it is taken from bone. So in from bone marrow, this diatom test is for diatom test, bone marrow is taken, or the sample from lungs, liver, or kidney it is taken. Um, for digestion, strong acid is used to dissolve the soft tissue. Then centrifuge is done to get the supernatant, and microscopic examination is done, and the types of compare the types of diatoms with sample water and suspected site. So as the we got the final diatom, it is compared with the suspected site where the body is or anything is drowned. From the sample water, it is compared, and it is the crime is confirmed. So next asphyxia is suffocation. This is a general lack of oxygen in the environment, seen in accidents, mine accidents, well, sealed room, etc. Suffocation, you guys know, we get lack of oxygen in environment, and suffocation is happened. So these called suffocation. Let's see medical legal importance of asphyxial death. This help in determining the cause and manner of death. Throttling and ligature strangulation is mostly seen in homicidal, and hanging is usually suicidal. Uh, choking, smothering can be accidental or homicidal. So in this case, we can find the cause of death. So until then, forensic medicine is done. Let's move on to biotechnology. Anybody from bio or biotechnology, can anybody say that what is the application of biotechnology that implied in forensic science and why it's used in forensic science? Anybody? Anyone? Hello, I'm
No, when the DNA has been found at the crime scene, whose DNA is that?
okay.
How
Okay,
the
I
modern
appreciate.
biology, how the body fluids like semen, saliva, blood, or any fluid related to body, how can it lead to the crime investigation, or how it can help in the further crime investigation? So overall study, you can say it biotechnology.
Okay. Sorry for the background noise, guys. So yeah, as you guys said, biotechnology is one of the very important role that is an important role in forensic investigation because it plays major role in it plays a major role in finding the in case of unidentified body or in paternity test or anything which related to crime. Forensic science only deal with finding the suspect. So biotechnology help in DNA profiling or in biological fluid identification, etc. Biotechnology plays an very important role. This enables detection, comparison, and interpretation of DNA, body fluids, tissue, microorganism to establish links between crime, suspect, and victim. So, as I said, these are the role of biotechnology, like in DNA profiling, human identification, paternity test, etc. So DNA profiling, this is the technique that used to find the regions of human DNA that vary from person to person. So because of this DNA profiling, we can find the, for example, in paternity or maternity test, we don't know that whether the person is, the pattern is suspected for the around the individual. So because of DNA profiling, the because of the DNA location around among the humans, we can find the suspected person around among the individual. So in this DNA profiling, STR plays a major role because the identification of the DNA, STR is seen, like for example, um, these are the small repeats that varies between individual. So these are one of the genetic markers. For example, TH01, it is the locus which is seen in the individual. We can find using it. You can see that this is the STR. So these are the short tandem repeats. So this will be vary among the individual. So because of this, we can find the suspected person. So these are the STR markers. So we can see that these things. and we can identify the, like, basically in forensic labs we will get profiles like this, like if paternity test, the profile of kid and the profile of suspected father, profile of mother will be there, and the profile will be match, like not match, it will be similar among the individual, like father, kid and mother, and we can easily find it. So PCR is the technique used in this. So you can see the samples is collected and the chemical method of DNA separation is done, and gel electrophoresis is carried out, and we will get the, these are the gels, we will get the this kind of bands, and we can find the, we will generate and profile using this. So let me hand over to Avishikta for the rest of the part.
So, Swapnil, can you please share the slide? Hello?
Yes, sure. Just give me a moment.
Okay, okay.
Can you please confirm if my screen is visible?
Yes, yes, your screen is visible. Can you share it from slide number 5?
Yeah. Just give me a moment. There might be some lag due to some network issue. Just give me a moment. This one, right?
From here it is not visible. It's still it is visible. So now I will continue from the blood, type of blood grouping and type of semen saliva test. So now can anyone say what is the introduction about serology? What is serology? Can anyone please say me what is serology?
Serology is a
Of blood
branch of biology
and
only. It deals
fluid.
with biological fluids.
Okay, so here is the introduction of serology, which is basically used as a individualization. Suppose there is a crime scene, so we can suppose there is a crime scene, so we can separate the person on basis of their blood grouping. If suppose A, B, and O blood group is present, so we can separate how many person was belong from A blood group and how many person belong from AB blood group or O blood group. So like this, we can separate the person from different blood group system. Can you please move to the next? Swapnil, can you please move to the next slide?
Yes, actually, I have already shared it. Please let me know if it's visible. I think there is some network issue.
Not visible. It's still stuck at
Just,
introduction part.
just a moment. Now?
It's now visible. So first is ABO blood grouping system. So ABO is classified like AB, and O blood grouping. So basically, it was given by first. It was discovered by the Carl Landstein. So who first give the idea about the ABO blood grouping. So based on antigen and antibody present in our body, so on that basis we have differently classified in blood group. Like A, if we go to A blood group, so antigen will be A and antibody will be B. And if you go for blood group B, then antigen will be B and antibody will be A. And for ABO blood group, is antigen is A and B both, and there will be no antibody present in AB. But in case of O, no antigen will be present, but antibody will be there. For antibodies, it will be A and B. So can we move to the next?
Hello?
It is not visible till now. Can you move to the next slide, please?
Just give me a minute. Actually, while I'm sharing the screen, I think the slideshow gets stuck for some reason. It's visible now?
Yeah, it's visible. So,
Okay.
as this blood grouping system plays a very important or very crucial role in the crime scene, as all the biological samples lead to a very unique identification. So, there is also one identification system is RH system. So, in RH is a blood grouping system determined by the presence or absence of RH antigen. So, here also it is determined by the presence of antigen and absence of antigen. So, like that, we can say if RH positive, the D antigen is present, and in Rh negative, then it will be the D antigen will be absent. So by this way we can determine whether the blood, what type of blood group is present. It's all about basis of antigen and antibody present in the blood. Can you move to the next, please? Swapnil, can you please move to the next slide?
Yes, it's visible now.
So like we have studied about the most of the forensic like forensic medicine. So if you come to the importance, like it's a very, it plays an important role. Basically forensic serology plays a very important role. Like it helps in the basically the main function with main importance thing in forensic science is suspect exclusion. Like as I already mentioned, like if this is a crime scene, we can separate the person on basis of their blood group, so it will narrow down our investigation process. Even blood stain comparison, we can also use the serological methods. Important in paternity dispute and medical legal cases, as my co-partner P has already described you guys ki how like how from a DNA we can use a single sheet of DNA can determine the paternity and maternity both the cases. And it also stable the it also give the resistance decompositions. Next slide, please. So these are the tests. There are two type of tests we perform basically. So presumptive test and the confirmative test. Basically, presumptive test are for blood. Basically, for every test, we first go for the physical or the basic test. Then we go for the confirmatory test. So for blood, we use basically Benzidine test, Castlemeyer test, leukocrystal violet test. Details the presence of, like, it's basically detect the presence of hemoglobin. So from blood test, we can also say that whether it is a animal blood or it is a human blood. Basically, this question first arises in every lab when it's any sample come, any biological sample come from come for testing. So first question the officer arises that whether it is a human blood or animal blood. So by performing the test we can say it. Like for confirmatory test we use Takayama, where we use Takayama reagents, and then it will form a brown crystal. So if we after performing the Takayama test, if we see there will be a brown crystal which presence, there is a presence of blood, and that is presence of human blood. And for Takeman, it's for brown crystal, and for Takayama, it's for pink crystal. So after performing the both the test, we can see two different type of crystal, which also confirm that yes, blood is present and it is a human blood. Can we go to the next slide, please? Swapnil, can you please move to the next slide? Okay. So these are the microscopic view of Takeman and Takayama. So in Takeman, as we can see, it's brown color crystal is formed, and for Takayama, it's pinkish color crystal is formed. So this is how it's look in for both the cases. This is how it looks under the microscope. So by seeing this kind of crystal formation, we can conclude that yeah, it is a blood present. Next slide, please. Okay, so it is a test for saliva. Basically, in rape cases, basically when culprit or the culprit used to get sexually get attached to the person, then some kind of saliva get transmitted to that victim's body. So, with the help of cotton swab or like that kind of things, we used to collect the sample, and then we send it for the analysis. So usually mainly in secrets for the status stage determination, about 80% of the individuals are secretes blood group substance ABH may be present in saliva. Absorption inhibition, so basically absorption inhibition test commonly used mainly secreted which are the secreted parts. So for we use the absorption inhibition method, and 80% of the individual are secretors blood group substance ABH may be present in the saliva. So for these both the things, absorption inhibition test is mainly or the very common test for saliva is absorption inhibition methods. Next slide, please.
Hello?
Yes, it's not visible. So basically, one of the another most important biological sample is semen. So basically, in case of sexual assault cases, we basically get the semen sample. So which also like helps to determine the cases. So basically, as we said that for sexual assault cases, we use semen sample as a very crucial, important evidence. So like acid phosphate test, Christmas tree test, confirmatory test. These are two acid phosphate and Christmas tree is basically for the presumptive, or you can say the primary test also. And for confirmatory, use microscopic identification of spermatozoa, or even you sometimes also in some cases we also use fluorescent tests and barbecue test. So as the this picture, this shows that how acid phosphorus test is seen under the microscope after processing all the acid phosphate tests. So these blue color crystals like these small crystals will be formed, not the crystal, this circles types things will be formed for the acid phosphorus test. And for the Christmas tree test, we use two reagents, and they get mixes with themselves with the semen sample, and they form a Christmas tree like structure and the color. So for this we can say that yeah, it is a Christmas. If the Christmas tree stain is coming, and this Christmas tree stage we only perform for the semen sample, because this is a very effective and confirmatory test for semen. Next slide, please. Next slide, please, Swapnil. Okay. So, what are the basically forensic investigations of both the samples? So, it's basically, as I said, ki it's very plays a crucial role in sexual assault investigation. As in case of sexual assault investigation, semen sample is very crucial. So, basically, it comes to our lab and we do the further investigation, the further analysis. It as the main thing of every sample is linking victim and accused. In case of bite marks also we can find the saliva sample, so we can also do the analysis on basis of that saliva sample. Helps in the suspect identification, as we already discussed. Next slide please. So here is come the main part of the documentation, as we can say that how the biological samples are collected from the crime scene, and how it packed and how it been transported or you transferred to the analysis or the lab. So this whole part is called the chain of custody. So as we went to this crime scene, so we have to first analysis the what are the biological sample were present. Then according to that we have to collect the sample, and we should remember every time while collecting the biological sample, especially you have to remember that it should not get contaminated or it should not get degraded, because biology sample we cannot get in a huge amount, it is very less. From that less amount we have to do the analysis part. So like gloves should be changed frequently to avoid cross contamination. Wet biological sample should be air dried before packing. So for the biological sample, if you get a blood which is wet, we have to wait for the air dry process. We cannot dry it, like we cannot use any other kind of drying method, hair dryer or something like that, heating process to air dry that blood. We have to wait for the air dry process only. When it will dry with the contamination of air only. So here is the collection. So blood stain are collected between, we have to take a swipe, and after swabbing it we have to sterilize, and then we have to put it in a EDTA vessel, and you have to send it to the lab. So for hair and fiber sample, we also have to use forceps, which should be clean. And by picking that hair or fiber sample, you have to put it in a Ziploc bag, and then you have to send. And we should need to remember first thing I'm telling you guys, as a forensic scientist, we have to remember that every packaging should be suitable for that sample. I cannot pack a biological sample in a Ziploc or in a blood sample in a Ziploc pack. So because that will get contaminated and it can destroy the samples. So we have to remember that which samples should be packed in which particular container or which particular box. Can you move to the next slide, please? Can you please share the next slide? So as I said, collection is done. So that are the collection process, and after packaging we have to always mention like case number, what is the number of that case, description of the evidence and what kind of evidence is present, whether it is blood, semen, saliva, or some cloth pieces or any fiber pieces. So always have to mention, so that the person who is accepting the evidence, they can understand that yes, it is a this kind of sample. We need to separate the sample on basis of that division. If it is written that biological sample or it is written that blood, so we have to send that sample to the biology division only. We cannot send the blood to the other division like physics or ballistics division. So these should be remembered, and these all the labeling should be mentioned in that packaging. So it is the main part of chain of custody. So as I said, this is a very crucial part, as the analysis part is also important. Before analysis, how we are packaging or how we are transferring the sample from the on spot crime scene to the laboratory, this whole total chain of custody will affect in your presenting the result in your courtroom. So if this if any misleading or any misguidance of sample happens during this packaging from collection of sample in the crime scene to the laboratory, so they have to give they have to face so many of queries where they have when they have to be present their report in the courtroom. So all the things should be remember as a scientist to maintain the chain of custody, which is very important. Can you move to the next slide, please? Uh, yes, so we also have to remember that collection and packaging also have done. So when we are coming this, like for in case of biological sample, we have to remember that we sample need to be, if there is any need, so like for freezing minus 20 to minus 80 degree for if we want to freeze something, then for DNA sample we have to maintain a temperature of minus 20 or minus 80. It depend upon that DNA sample. For some kind of sample also there which we need to refrigerate so that it do not get decomposed in when it come in contact with the laboratory environment. So even EDTA is also used for blood storing, and so you have to maintain a proper. Not only that, we have to also maintain a proper temperature of the lab. Like for there is a in biology section, I don't know how many of you visit biology section in any lab. CFSL or FSL lab. So in that case, you can see if someday you can visit, and you can see there is a separate temperature cabinets for every biological sample, for DNA, for blood, like for other biological sample also, if they need the refrigerator, so there is alag alag the different temperature storage cabinet where they used to store the biological sample so that it can maintain its so that it do not get decomposed or do not get harm the sample. As I already mentioned that biological samples are, when you collect it from the crime scene, they're in a very minimum amount. So by that minimum amount we have to do all the steps, and we also have to make the reports. We have always have to remember that how can we preserve that, and from a small amount how can we do the further and all the processes or all the analysis for that biological sample. Next slide please. Okay, so I will hand over next to the Priya.
Has my screen sharing stopped? Can anyone please confirm if my screen sharing has stopped?
Yes, Swapnil, it's stop.
Okay.
Yes, sir.
Over to you, Priya.
My screen is visible.
Yes, Priya, it is visible.
Yeah. So yeah, let's see the application of forensic biotechnology in the field of forensic science. So forensic biotechnology is applied to analyze biological and evidence and establish identity and relationship in forensic cases. It supports criminal investigation, legal decisions, and other things. So basically, briefly, we will say that forensic it helps in identifying the biological evidence, as Avishikta said, and it also helps in identifying the relationship in the sense, as I said before, paternity, maternity, or in the unidentified body also. It is used, and it is help the individuals to find the relation of the unknown person. So the key application is suspect identification. So in the DNA profiling, uh, the suspected per individual, the samples like blood, saliva, semen, hair, skin cells are collected from the suspected or crime scene. Then DNA profiled are made and compared with suspects. This help in link a suspect to a crime or eliminate an innocent person. Paternity kinships testing. So this intercept DNA comparison between individual determines biological relationship. This is called paternity and kinship testing. So in this, used in child custody cases, inheritance disputes, and confirming family ties. Ties. So in this case, biotechnology is used identifying missing persons and disaster victims. DNA extracted from bone, teeth, or tissue is matched with relative or stored database. This is crucial in mass disasters, accident, unclaimed bodies, long-term missing cases, etc. And yeah, so today's lecture is done for the day. So you guys can ask if any questions. Any questions from my side, Avishikta and Jyoti? You can ask if any questions or any query or regarding anything, you can ask. Hello. Participants, am I audible?
Yes, you're audible. So I think there's some queries in the chat box. You can watch them first and maybe reply to them. So I think there is a query from Disha. If a killer causes a sharp force injury, then stitches the wound after death to hide the crime, what will be the things the experts will analyze? You can open the chats maybe. Speakers are requested to please open the chats and analyze them.
I'm having some network issue. Can can you repeat the query asked in chat?
Just give me a moment. I'm sending the queries to you. Meanwhile, Abhishekta and Jyoti, if you guys are getting any queries, please address them.
I got one question. Any color is observed from Avanika Verma. So, any color is observed while performing any of the preliminary test of semen? Yes, like a grayish color with a light greenish shade is seen while we perform the test or the preliminary test of semen. On rest for confirmatory, or if you want to know whether it is semen sample or not, you most of the time people used to do, or the laboratory person used to do that Christmas tree test, which is basically very useful for determining one shot determining the semen sample. Thank you.
So regarding the wound, may I know, is the stitches is are you saying that is the stitches is made right after the uh, crime, or are you asking like after, if the crime blunt force like sharp force injury is done, uh, right after the injury the stitches is done? Can you just say that, confirm
Right
me
after
it?
the injury is caused, the killer tried to stitch the wound so that the crime can be hidden from that in that case.
Okay.
So one more question is there. Oh, sorry.
Yeah, it's fine, Avishikta.
Okay.
So, like basically, if the killer is doing the sharp force injury and he or she is stitching it to, uh, like to not to find it, we can see if the bleeding, if the suspect or if the killer is done it and he is wiping the bleeding and he is stitching, means the reaction in the wounds, like if somebody is stitching it, we can see the bleeding into the tissue or the inflammation, that proves the injury before that, right? So if somebody is stitching, we can see the inflammation, and it will show it's done, whether it's before, right after the sharp force injury is done, or it is done before. So the inflammation is seen, and we can find it.
I got one more question from Niharika. As it said that if it's semen, then what is observed in Christmas tree test, and if not, then what? So as we can say that if it is semen sample, if we perform the Christmas tree test, we use basically nuclear first red and picrocarmidine. There are two type of markers which use. So after putting these two markers, we can see that the head portion of the semen, it appears to be red or pinkish, and if in the tail portion we can see that green or blue color. So by this we can confirm that it the sample is semen sample, and if that is not semen sample, then we can only see that epithelial cells or some kind of cells which are present in saliva. So by this way we can determine whether that sample is semen or not. Thank you. I got one more question, guys, from Disha. Like, how would you analyze semen collected from a dried piece of cloth, and what test can be performed, and what precautions are required? As we can perform a visual analysis test or a ASL test, and then we have to go for the presumptive test only. And like with the experience, we can get to know that whether which part of the cloth is basically having the semen sample. Most of the cases they send the undergarment, so from that it's like not too easy, but it's little bit easy to identify whether which partner. Yes, we can obviously from dried blood cloth we can go for the semen sample analysis. Precaution, we have to see whether that the cloth how they are packaging, because sometimes police officers don't pack it in a good way and they send the sample, so there is a high chance of getting contamination. So yes, after coming to lab we have to maintain some precaution measure, like to save that not getting to more degraded.
Any other questions regarding forensic science, biology, or anything regarding the handling, packaging, or things happening in forensic labs? If anybody have any queries, let me know. We will solve it.
I think one more question is Sarvan Ananda, like 2006 at the gmail.com. So as a person is saying that just by examination of bloodstain pattern can you determine the cause of death, ma'am? No, we cannot actually determine the cause of death from the bloodstain, but we can, by analyzing the bloodstain pattern, we can say that what kind of weapon is used. We can like if we shoot by a gun, if a person is shoot by a gun, then the blood spatter or the bloodstain pattern will be different. If the person is murdered by a knife, then that will create a separate bloodstain pattern. So by analyzing the bloodstain pattern, we can some extent can say that what is the instrument or what is the use for that murdering of that or for killing of that person.
Okay, let me just give a brief how the reports are written while, like, after conducting a full biological examination of anything. So firstly, we will get the samples like exhibits. We will get it from the police station to the lab. So it will be basically, it will be parcel the, as you guys have been studied, or if you don't know, bloods and urine sample, basically it will come in vials for toxicological examination and for DNA examination as well. And the semen samples we will get in slides. If it's any assault case or any suspicion, the whole, if for example it is a sexual assault case, the exhibit, the police or any other who's collecting the exhibits from the crime scene, they will send the whole, for example, if the semen stains are seen in an underwear, they will send the whole underwear to us, and we will cutting the small piece, and we will be having it as an exhibit. And swabs like blood swabs and anal swabs and some case swabs will also be sent. And while getting the exhibits, there, and one assistant will be there basically in a forensic lab. So the forensic officer, he will open the exhibit. Right after opening the parcel, there will, if it is a sexual assault, there will be a sexual assault kit. The exhibit itself will come as a kit, like sexual assault kit. So we will photograph that kit using an ruler. So this is the purpose for the documentation in the courtroom. For the legal purpose, it is documented. Each and every step is documented. So after, if the sexual assault kit box is getting, they will measure it using a ruler, and as the ruler is there, they will take an photograph. And the sexual assault kit box will have the kits like swabs. Two swabs will be the blood swabs, anal swabs, semen swab. And then if there any suspected things like clothes, bed sheet, underwear, hair strands, anything will be there, they will send it to us. While if we are getting a blood, we should not write, and it is not written in the report like the officer, crime scene officer is sending and has sent blood to us for examination. It is not written like that. How the report is documented before taking an exhibit for DNA examination or any other examination, it is also write as a written form. So if they are getting a blood and vial, they will write that, and brown color liquid mentioned that blood sample of the suspect has been received. So they will write this thing only, they don't write blood. And if they are getting semen swab or saliva swab, they will write that there is a glass swab, written that swabs of the suspect, swabs of the victim. We can't confirm that it is a semen swab, it is a saliva swab. So they usually don't use to write it like that. And if the hair strands are there, they will write two strands of hair, fiber, a string of fiber is there, a cloth piece is there, and undergarment is there. So like this, they will write the report of the exhibits, and they will take the small, as Avishikta said, that we don't get the samples as in large quantity. If if there are small drops of blood also, the crime scene officer who's taking evidence from crime scene, they will send the whole bed sheet. If they are seeing a paint stain or any other stain in one cloth piece, they will send the whole cloth piece. So we need to take the cuttings of that. If they are sending blood, we should take small quantity of vials from the blood from the vials. And we should, the samples are not thrown after getting it. It is safely, it is packed, and again it is sent to the police station. If we are getting blood, fetus, or semen, anything, the blood and fetus it is definitely stored under the control conditions. And this is carried out through the DNA profiling, like a PCR is done, then gel electrophoresis, then like quantification of DNA is done, then the profiles are generated. At last, again, the forensic officer, like a JSO, like anybody forensic officer who's taking that case, he will write the results which he got. If he is getting any DNA profile from the blood. He will be writing that exhibit number 1, red color, brown color liquid, it has this DNA profile, and they will write like this. And uh, they will write the penal code and all in a yellow color envelope, and they will write the case details, IPC, and in which section it comes under, which police station it belongs to. They write all these things, and they will pack the balance what exhibits are got. For example, as I said, the cuttings of bedsheet, the small spots are only taken for DNA examination. Balance things will be there, right? So these things are uh, put in that envelope, and it is sent back to police station. So these are the steps that are carried inside the forensic lab after receiving the exhibits, parcel exhibits, and they will send it as how it got safely. They will send it back to this is the things happening in forensic lab. If anybody have any doubt, you can let me know.
Thank you so much, ma'am. So now I suppose that you guys are not having any further doubts, because I have seen that since we are running short on time and the session has already been going on for the last 2.5 hours, I can say, right? Or maybe even more than that. So that's why I tried to answer the questions as quickly as possible in the chat box. I'm really sorry, speakers. However, if you want specific doubts to be addressed by the speakers, then guys, you can reach out to them via their email IDs or their LinkedIn profiles. I would definitely share them in the group once the session is over. Thank you so much once again, speakers, for this delightful session. Now we would quickly move on to the vote of thanks part. But before that, I would just like to ask the speakers if they have prepared any specific assignment for you guys. To you, Priya.
Yes, as me and
Like,
Priya, okay, as
Yes,
me
please.
and Priya
yeah,
have
me and
decided.
Avishikta, yeah, we have decided that participants can take any of the topics we have took today. They can either make an PPT or a flyer. Then we will decide among the participants, and we are thinking to post in LinkedIn along with the profile. So
Great,
you
great.
can either make PPT
So the
or
best,
flyers regarding this topic.
got it. So the best posters will be featured in the official LinkedIn page of Forensic Fusion.
Yes. Or
Okay.
I was saying that
Yes.
it could be a poster also. Any topic related to what we have taught today in the lecture, or anything related to forensic medicine or forensic serology or biology. Any topic you can
Great.
make any poster or any PPT. So the best PPT or the best poster we can post in our social media accounts.
Great. So to just make things clear, guys, you can prepare any flyer, any poster, if you want a series of PPT, let's say, but not more than 5 slides, I suppose. And other than that, you can also make flowcharts if you would like. Okay. You can either make it by hand if you like painting and stuff. You can either make it on digital platforms like Canva, or you can also use AI tools if you would like to. But we really value your creativity, so please ensure, even if you are using AI, do not entirely rely on AI, right? Make it yourself rather. So thank you so much once again, guys. Thank you to our incredible panel for such a detailed walkthrough of the biomedical field. Miss Priya, your breakdown of DNA extraction and serology was masterful. You showed us that biology is not just about blood typing anymore, it is about extracting identity from the smallest trace of saliva. Miss Jyoti, thank you for taking us inside the toxicology lab. Your insights into the organophosphorus poisoning and postmortem observations bridge the gap between the chemical reaction and cause of death. and Miss Avishikta, thank you for clarifying the procedural part. Understanding how to preserve viscera and write a post-mortem report is what saves a case from falling apart in court. And you explained that beautifully. To our interns, today you learned how a body talks through its DNA, through the poisons in its system, and through the medical examination. You would soon get the PPT for today's session after the session ends, okay, in the WhatsApp group. Also, we will meet again tomorrow, and tomorrow we are having an interesting session on body behavior, and we are having an international expert joining us from Lebanon. So I would request all of you guys to be present in the meeting. Thank you so much once again, guys, for being an incredible audience and for staying till the end. Thank you so much. Have a great evening ahead. Bye bye.
Okay.